Hi, everyone. Thank you for the kind introduction. And my name is Miriam Poley. I am first year PhD student in computational biology and biophysics at the Barcelona Supercomputing Center in Spain. And I'm here today to present our ongoing project, which is called Exploring the Biophysical Boundaries of Protein Families with Deep Learning Methods. First of all, we started this project because we want to understand why natural protein sequences are as they are nowadays and how deep learning models could help to understand this regard through evolution. These models are basically algorithms that allow the design and generation of protein sequences by analyzing and extracting information from real proteins. So they suppose to learn patterns and features present in real proteins in order to make relate predictions when inferring. In the last year, all these models have been classified in so many ways, depending on the problems they try to solve, but we focus on models for reverse folding, like, for example, protein NPNN or PROST5, and models for sequence generation, like, for example, ProGPT2. This thing of reverse folding is about using 3D structures as input to generate protein sequences that fall into the given structure. That means that we can generate protein sequences with a similar folding, which is basically the opposite of what AlphaFold does. While models for sequence generation need to learn the grammar present in protein sequences and basically how to generate protein sequences in a specific protein context. For that case, in most of the cases, the fine-tuning is what allows to define exactly the space that we want to explore. But from a biological point of view, all these models have something in common, and it's the lack of interpretability. We need to understand and interpret what these models are actually generating. For that reason, in this work, we are proposing to measure the energetic of artificial ensembles, but how? We propose to use local energetic frustration. This concept comes from the fact that proteins are not only optimized for folding and stability, proteins are also evolutionarily selected to function. And this could explain that almost the 10% of the internal interaction in proteins are in energetic conflict with their local structure. And these energetic conflicts seem to be preserved over evolutionary and physiological timescales, making possible protein functions. The good thing is that we have a tool called frustratometer to measure this energetic conflict. Basically, a frustratometer measure how well optimized for folding the energy of a given residue or interaction is in comparison to the random variance that we can sample in non-native conformation. So with frustratometer, we can classify all the residues in a protein structure in three different frustration states: highly frustrated, minimally frustrated, and neutral. Being the highly frustrated less favorable for the protein energetic and likely related to functionality, while the minimally frustrated are the more favorable for the protein energetic and likely related to stability. But statistically, it's difficult to interpret frustration on a single structure. For that reason, we are including conservation, and we are developing our tool FrustraEvo to measure the conservation of the local energetic frustration. So with FrustraEvo, in the same way, we can map frustration from a group of related structure to a multiple sequence alignment to have what we call a multiple sequence frustration alignment, and then check using information content how conserved its frustration state and sequence identities at its position on the alignment. So all this information can be summarized in this kind of logos, where the size of the letter and the age of the bars are related to conservation. The bigger, the more conserved. And again, the colors are related to the three different frustration states. So using this information along any protein family, we can define a residue level for different scenarios when frustration and identity are conserved, when just is conserved the one or the other, or when any of them is conserved. And this is basically what we did with native protein families for example, in beta-lactamases, an example of enzymatic protein family, and alpha globin, an example of protein-protein interactions. And we found that there is 3 and 12 highly frustrated and conserved positions in these families. And in the case of alpha globin, most of them were related to protein-protein interactions, as we expected. And in the case of beta-lactamases, um, all of them were annotated catalytic site according to the literature. So the question now is if these models are able to recover this highly frustrated position, and how similar they could be compared to the natural ensembles. So if we generate protein sequences using these models, protein NPNN, PROST5, or ProGPT2, we predict the structure using, for example, ESM-Fold. Then we can apply the same frustration analysis and compare the energetic signature in native and artificial ensembles. But the problem here is this a lot of information, and now it's not that easy to look for these patterns. So we are introducing consensus sequences in such cases, where again the site of the letters refers to the sequence identity conservation, and only the positions where a conserved frustrated state is colored. But again, the 4 different scenarios are the same as before. Let's start with the result of artificial globins by ProGPT2 once we fine-tune the model. Here in the logos, we have selected the 12 functional sites annotated in alpha globins, and also just below for the full comparison with the ProGPT2 ensembles. And as you may notice, the amino acid distribution is very close to the native distribution, but also the frustration states are quite similar in between. In fact, all the functional sites are fully recovered in frustration and identity, but in ProGPT ensembles, new highly frustrated positions appears. Almost the same situation is that we notice in artificial alpha globins from PROST5, even when we didn't perform fine-tuning or retraining at all. In this case, we recover most of the identities, but not always the conservation of the highly frustrated state is recovered. But in the case of PROST5, we also obtain new highly frustrated positions. The thing is that for now we still don't know the functional implication that these chains proposed by generative models may have in the protein energetic, but it's quite surprising also what happened with models like protein NPNN. In the case of protein NPNN, this is a graph neural network trained with structural data. And in the case of alpha globins, the model doesn't recover any of the functional site annotated in beta-lactamases. Even in some cases, the functional sites are substituted by less frustrated amino acid, as is the case of glutamic 27 or the lysine 99, resulting now in a disagreement with the frustration pattern in native globins. This could be a consequence of the sampling temperature, because we are generating sequences with temperature 1, and according to protein NPNN, the amino acid bias, the amino, the amino acid bias is a consequence of the sampling temperature, in a way that at higher temperatures, the amino acid bias decreases, producing protein NPNN ensembles that are less stable than the native ensembles of alpha globins. But however, we check all the temperatures, and this functional site, this highly frustrated functional site, are not recovered for any temperature, maximizing in all the cases the protein stability. But however, results become more interesting when we analyze beta-lactamases, because in this case, protein NPNN is able to recover most of the catalytic site, even when some of them are unfavorable for the protein energetic, because they are highly frustrated. In fact, if we check again all the temperatures, we notice that these highly frustrated catalytic sites are recovered in all the cases, and the amino acid bias also remains. suggesting that there is a strong bias toward the native identities, but more specifically towards the native identities of functional models, since protein and PNN ensembles are now more similar to their native one. That means that even though functionality and stability are sometimes in conflict, protein and PNN is not always able to remodel the functional side to gain stability. So at this point, our hypothesis here is that there is maybe a consequence of the intrinsic evolutionary information present in the training data set. So to try to prove it, we are mutating in silico all the catalytic sites present in native beta-lactamases to a different amino acid involving a minimally frustrated state, for example, to valines. We have checked that these valines are minimally frustrated compared to the native identities of the catalytic site in native beta-lactamases. So we decided to use this mutated variant as target for protein and PNN instead of the native target. However, we obtained again that reverse folding with protein and PNN using these mutants and relaxed mutants still recover the highly frustrated and conserved catalytic site present in native beta-lactamases. So what happened, this is consistent with our hypothesis, but what happened if we retrain protein and PNN, removing from the original dataset all the non-enzymatic data? This is supposed to remove almost the 50% of the original data, and is basically what we did. And we have a very, very preliminary results suggesting that at least the identities of some of the catalytic site of native beta-lactamases are recovered even when the model hasn't been trained with enzymatic data, which is not conclusive for the moment, but at the same time make us think that perhaps there is a structural predefinition for the functional site in proteins. This is something that we will keep investigating for the community, but so far we can conclude that the artificial protein design is consistent with the native energetic frustration, even when these models have not been trained with frustration data yet. That deep learning-based protein models are really useful for revealing inherent patterns in the evolutionary history of protein families, but especially reverse folding algorithms, because they allow us to sample how much variability the same fold can present, but still recovering functional residues. And finally, that for sure, combining this algorithm with simple interpretation tools as frustration could be a powerful way to discover and annotate unknown functional residues. So just before finish, I would like to say thank you to my team, also to my collaborators, and thank you to you for coming and listening.
We have time for questions. Hi, Miriam. Nice work. I got lost a little bit in how you found your examples, the beta-lactamase, and I don't remember which one was the other one. If it was like a wide net, and then you chose the most frustrated ones, or what, you were working on those particularly?
No, we are using these protein families because they are model families that have been studied in the group. And we also have recent papers measuring the frustration in the native families. So it's a way to start with the comparison in well-known and well-studied protein families. But the idea is to expand the analysis to all the protein families to have like a general idea of what frustration is being recorded in artificial ensembles.
Okay, and my second question with beta-lactamases, you know, antibiotic resistance, right? So you have new antibiotics, they mutate, and then they recognize. So does the frustration change with the substrate?
How the frustration changes with the
change in antibiotic recognition, right? So if you go from penicillin to some of the other derivatives.
I don't know how it's changed because we are now just comparing frustration patterns, but the idea is also to test some experimental design with our collaborators if we can, just to check that this change could be also functional.
Okay, thanks.
Awesome talk.
Thank you, Jana.
Not sure it's a question to you, but you may know the answer. And if not, a question to you. Have you looked at families of proteins that exist across temperature scales in bacteria, for example? So everything from psychrophile at low temperature to thermophile, high temperature, would probably display different patterns of frustration, right? And could somehow answer some of the questions that you've been talking about.
I don't know. We need to try different families, and especially enzymatic families, because we have tested this approach only in beta-lactamases, but maybe the situation is different in other kind of enzymatic cases.
But do you expect that frustration signatures or patterns are going to be different across temperature?
Yes, I expect that, but I'm not sure now.
Okay.
So thank you for your talk. I have a question regarding you have a 3-level classification, high, medium, low, right?
Yeah,
So
for the frustration.
And the frustration. So are you thinking about kind of using more continuous values to explore, more like the boundaries of these classification things? Because maybe you will find some.
In one of our last papers, we also include the median, that is like measure the frustration, but at a continuum scale. But the problem is the limit when the frustration state is considered or not. So to define exactly the pattern, we are now using this approach, but maybe we can think about it for the next analysis.